Okay, great. So thanks everyone for coming. It's great I get to introduce my colleague, Claudiu Gradinaru here. Just his lab's around the corner from me. Claudiu's lab focuses on single molecule fluorescence imaging and spectroscopy. And so for those of you who aren't familiar with that, biology for its history has basically studied the biological world by looking at test tubes full of molecules. And then the physicists kind of got in the mix here and decided to look at individual molecules, which you can imagine actually trying to do that is massively more complicated. And that's exactly the sort of work that Claudiu does, is trying to look at these single molecules by themselves. And from that, the range and richness of what you can actually pull from the data is so much more than you'd ever get out of a test tube. Okay, and so I'd say Claudiu's lab has been focusing for a number of years on two very challenging problems in the world of biological physics. One is studying what's known as GPCRs, which are called G protein coupled receptors. So they're a massively large class of membrane proteins. About 40% of all prescription drugs target GPCRs. And his lab looks to study nanometer scale conformational changes that can often lead to nanosecond type dynamical reorganization of these proteins. So they're signaling proteins, and how they get turned on and how they cooperatively interact and form large networks of oligomers is really one of the big questions in the field at the moment. And the hope is that by understanding this new biology, you can generate all sorts of new drugs, or at least new approaches to drug development. Today, I believe Claudiu is going to be talking about basically the second half of his research program, which is studying something known as a intrinsically disordered protein, or an IDP. And so what his lab does is they generate the single molecule data, they combine it with structural data from other techniques like NMR, X-ray scattering, and try to get insight into this very funny sort of protein, which is intrinsically disordered by its name. But what does that mean? So most proteins, they form and they fold into a very controlled structure. And this protein folding problem is like one of the big problems out there to be solved, which perhaps has been solved by AlphaFold or perhaps not. But that's not the end of the story. There winds up being an entire range of these very odd proteins that don't fold into a predefined conformation, rather are intrinsically disordered and yet have functional significance that we're only at the moment being able to get some sort of a handle on. And so with that said, I think that should give you a good background a little bit. It's a great honor to pass this over to Claudiu.
Thank you, Josh, for the kind introduction. Thank you to the committee for inviting me to give a talk. It's been a little bit over 15 years since I joined UFT, and I hope to give you, my colleagues, and the students in physics a flavor of what biological physicists, biophysicists do when they have fun in the lab. I'm an experimental biophysicist, but the pandemic forced me to do some thinking about what do my data mean in the larger context. So as I think Josh mentioned, I will be happy to answer your questions at the end. Please post them in the chat or in the Q&A. I hope not to bore you for the next 50 minutes or so. Just want to give you first a background, the flavor of what sort of research themes are or have been going on in my lab. I'm a spectroscopist. We do look, as Josh pointed out, the two major classes of proteins. This one I'm not going to be talking about today, GPCRs, G protein coupled receptors. Whenever you take a medicine, chances are that some receptor in your body is the target of that medicine that the doctor prescribed you. On the other end of the spectrum that we study is IDPs, or disordered proteins. I'm going to be talking about these today. They have been labeled as the dark matter of biology for a number of years because they escape traditional ways of describing them. Since you can't crystallize and you don't stably form crystals, then it's very hard to get these beautiful 3D structures like you see for GPCRs. Since I'm in a chemical and physical sciences at UTM, chemists are always knocking on my door, and I've been collaborating with a number of them throughout the years on everything from biophysical assays to test new cancer therapeutics, to nanoparticles for biosensing or for imaging and therapeutics, or to push the limits of bioanalytical applications, detecting, for instance, the low number of copies of DNA or RNA in a single cell. But today, I'm not going to talk about all these themes. I want to give you first a flavor of what sort of instruments we built in the last 15 years in the lab. By the way, I've been blessed with great graduate students, exclusively physics graduate students that overcame their fear of biology and probably they were attracted first by physics and then they stayed because of biology. But these are we call them microscopes, but you can't look through an ocular in this microscope, these black boxes that are there. And they're nothing else than laser light that is modulated and shaped and change colors to excite our molecules of interest. And once that happens, the fun begins, because what we have, particularly in this setup, which is one of the 3 microscopes in my lab, we detect signals from single molecules in solution. They appear these bursts of light, and we use this avalanche photodiodes, which for each photon detected output a TTL pulse. And basically the data here is a list of photons with polarization, color, time tag, and so on. So you can do all kind of interesting multi-parameter data mining in this multidimensional space that the data offers. So both Baoshi and Greg spend their best time in the lab building, designing, rebuilding this setup. And another grad student, Amir, there is also some signal in the noise. So there is, even if you don't go all the way down to single molecule, but just a few molecules, in your observation volume, you can look at the fluctuations of your signal, and you can, by building this time domain, so-called autocorrelation of the signal, you can determine different dynamics in your molecule of interest. For instance, what's highlighted here over 8 orders of magnitude in time, from 1 nanosecond to about 100 milliseconds. You have diffusion, have photophysics, you even have anti-bunching when the nanosecond timescale. So you can use this, particularly this part here, the photophysics, in the microsecond timescales to look at normal proteins. This is a GPCR, by the way, a receptor. And Dennis, another recent graduate, used this dynamic quenching. Our probes get quenched by natural amino acids inside the protein of interest. And this can report on the normal dynamics of normal proteins. Even when you see such a beautiful structure, it's not static. So what Dennis was able to distinguish is how this dynamics of this one of these helices was modulated by different drugs, agonists, antagonists. So the kind of drugs that you take, this is adenosine receptor. So when you drink coffee, this receptor reacts, by the way. So you may change some of these fast dynamics in the receptor. This is called photo-induced electron transfer for some of the specialists in the audience. Another one of my grad student, postgrad, Yu Chong, he took the fluorescence correlation spectroscopy and he said, what if I shine 2 lasers at the same time in a tiny volume, and I look at molecules with different tags if they're cross-correlated? So this is basically the Einsatz. You look at noise in 2 different channels, and then you cross-correlate these fluctuations. And if you find a non-zero amplitude, that's something that tells you about the complex that these 2 species can form. What is the percentage of molecules that form a complex? So this is great if you want to go into live cells, for instance, and you want to distinguish something that is stochastically co-localized here from something that is really correlated. So in this particular case, these 2 molecules, they have different tags. You see a green and a red tag. They form a complex. This is a partner protein of the receptor called G protein. And when they move around in the cell, in this Brownian or quasi-Brownian trajectories, they remain correlated, and you see the high degree of cross-correlation. Here, although the signals are co-localized, you see the green and the red pretty much at the same spot. These two proteins, they are stochastically moving around each other, so there is almost zero cross-correlation. So it's a great technique to capture this complexation of biomolecules within the native environment. The last little gadget that I want to show you is it's using evanescent field illumination. It's called total internal reflection microscope. The latest victim that I recruited in my lab, Xiaohan, he's now optimizing and further improving this machine. You can see again several laser sources, and acoustic optic tunable filters, different EMCCD cameras. And here the sample is sitting on a stage, it's temperature controlled and so on. What can we do with this kind of setup? Imagine that what you're basically looking here, you're looking from the bottom up. So these are particles, molecules that are immobilized on the glass surface. And the evanescent field allows you to see what's within approximately 100 nanometer deep into the solution. So what we do with this, by looking at the brightness of the spots, we can count the number of steps, get histograms, do the statistical analysis, and infer what is the degree of oligomerization of the pores. A lot of these receptors or channels or pores, they form supramolecular complexes. Of course, this is an isolated form, it's not in cell, but here you can look at a live cell. And again, you're looking from the bottom, look from the bottom up here. This is the bottom membrane of the cell, and you can see all kinds of particles, whether they're single molecules or aggregates, or cargoes moving along by some filaments, actin filaments in the cell. You can analyze the diffusion patterns of this biomolecule. All right, so, okay, this is enough of gadgets. Let's get more serious a little bit about why would one, in any healthy state of mind, would want to go single, like a single molecule. Why would, the biology is complex enough to control, to measure. Why would you have to fight all these battles to go single? It's because in a nutshell, biological samples are intrinsically heterogeneous. So even if we don't talk about disorder, and we'll get there in a minute, they exist in various states, thermodynamically stable states, bound and unbound, open and closed, aggregated, folded, unfolded, amyloid. So if you think of amyloid diseases, this is highly relevant. So in order to be able to distinguish between this any number of states that are dynamically interchanging, we don't have the luxury to do what ultra-fast laser physicists, like I was one of them about 25 years ago. We can't have a pulse, a laser pulse to synchronize all our molecules in the cell. So to have everything act in sync. So what the other way to analyze it is to isolate them and to look at one molecule at a time. And with this, I want to say something maybe for the students in the audience, probably the other ones are already familiar with the fact that averages can be misleading. And it's not only when you do social studies or polling, but in this particular case, if you look at two states and if you do a measurement of this protein in a test tube, the average observable doesn't correspond to any physical state. The other stuff that I want to mention is even when you don't have such a 50-50 population, diseases often occur as rare events. It's an outlier, and in order to detect really minor populations, you need a technique that is able to resolve them in a context of a wider distribution. So my favorite argument is not here. The argument why I like to use single molecule is not to resolve heterogeneous population, although it's a strong one, is because of dynamics. I look at a single runner on the track. I don't have 10 asynchronous runners. I alluded to it about ultra-fast laser syncing. So technically, there are some challenges to this. So what does one have to do in order to go single molecule? Well, we impose that in our detection volume, which is, by the way, in the order of a femtoliter, think of it about a cubic micron. When you focus the light to diffraction limit spot, the lateral dimension here is a few hundred nanometers. This is maybe 1 or 2 microns. So the condition, if you impose that in 99% of the case, you have one molecule, where the probability of having 2 or more is less than 1% of probability of having one, in the Poisson statistics, you can infer that the average number of molecules in this volume is 0.02, which gives you a ridiculously low concentration of material that you want to study. So you have 50 picomolar of material. So most of the time, when we look at our single molecules, we see nothing. There is just the background. That's why our detector have to have a low noise, low dark counts. So occasionally when a molecule crosses this volume, it takes about a millisecond of diffusion time, the kind of molecules that we study. You see these bursts of photons, and don't be fooled by, these are not negative bursts. They are 2 different spectral channels, and they're plotted pointing in different directions. But we run programs that are identifying bursts, classifying bursts of having only one color or both colors. And typically the bursts have at least 20 photons in that. So a measurement that we perform typically acquires a few thousand such bursts, and then we do our statistical magic on it. So the technique that I'm going to allude mostly in this talk has something to do with dipole-dipole energy transfer. And Theodore Forster in 1946, I believe, came up with a theory to explain it. Think of it as the dipole interaction is 1 over distance to the 3rd. If you take the probability is 1 over the distance to the 6th. And that's what you see here. The efficiency of transferring energy, if you pump energy into the blue fluorophore, the probability of transferring to the red fluorophore goes as this distance dependence. And as the luck has it for current labels that we use in our methods, the sensitivity range happens to be between approximately 3 and 8 nanometers, which also is the relevant length scale of size of proteins, size of protein complexes, and so on. So what I wanted to say is that once we get all these bursts of fluorescence, we count the photons in each range, donor or acceptor, we call them. We calculate and we correct this fret efficiency, this ratio. Then we get this histogram. We live a lot on the histogram space, either unidimensional, multidimensional histograms. And as you can see here, you have already capturing some heterogeneity in the structure, conformations of this protein. I just want to draw your attention again. If you do a test tube measurement in a lab, in a spectrometer, you would get an average efficiency of about, say, 60%, which doesn't correspond to any or to very rare physical state. It would give you a false information. Right, so I think that's enough for the first, 15 so minutes of introduction. Let's talk about the object of my study, of our study in the lab. So different kind of protein you're probably used with these helices or beta sheets. This is our normal folded protein looks. They're obtained by crystallography, electron diffraction, and so on. So it relies on pretty tedious work to get this, nowadays even cryo-electron microscopy. So you often have a static picture, and typically you get some clues by looking at the structure of how the protein works. Around 2.5 decades ago, a new paradigm emerged in the field of intrinsically disordered proteins, which don't have a single stable structure but an ensemble of conformations. And believe it or not, although it was initially discarded as fantasy, disorder has an implication for function. Disorder doesn't mean lack of function. It's one, if you would take this protein and boil it and unfold it, it would lose its function. So that's why people are biased towards lack of structure, lack of function. So in biology, nowadays people think, the current view is that there is an entire spectrum of protein structure from all rigid, quasi-rigid structure to little flexibility. Then you have all the way to super disordered ensembles. You have domains, you have certain folded coexisting with unfolded domains. And you have a multitude of functions, such as disordered proteins that switch from one folding to another, or from transiently from one folding to another, or they act as mortar or epoxy joining two different folded proteins. So the kind of proteins that I'm studying fall into this category, so-called fuzzy complexes. So if you put your thermodynamics hat for a minute, and you remember that, I mean, a folded protein, think of it as a very well-defined deep well on the free energy landscape, or say enthalpy winds. All these molecular, intramolecular interactions win over the multitude of entropic states out there, the unfolded state. Now, a disordered energy landscape looks quite a bit different. It's relatively flat, but not featureless. So you have this rugged energy landscape, and one can ask what conformational features are relevant to functions, because this is a physical representation where you have a different balance between enthalpy and entropy. And one would ask, is each of this local minima relevant for function, or only some of them, so on. So the outline of the talk that I'm going to go from the next half an hour or so is that the conformational ensembles of disordered proteins are optimized from function. And there is a formalism of disordered chain called polymer physics. And what we want to look at is, using that as null hypothesis, what stands out in the context of homo or heteropolymer for these disordered proteins. I'm going to talk primarily about SIC1, but if I have a little bit of time at the end, I'll touch on a even weirder disordered protein called 4EBP2. Forgive me for these acronyms, these three-letter words where letters and numbers, biologists use them all the time, and you probably get tired or bored of them, but let's call them A and B. What do we require to arrive at this ensemble description? So instead of folded protein, single structure, disordered protein, finite number of conformers that are consistent with all the experimental observation, and hopefully they can explain the function of this disordered protein. So we need some experimental techniques that are sensitive to this heterogeneity. And we also need a modeling approach that doesn't really simplify everything to homopolymer physics. So let me tell you in one slide, what does this protein SIC1 do? It's a gatekeeper of the cell cycle. So if the yeast is not mature enough, SIC1 says, no, you're not going to go from G1 phase to, so it's like a maturity clock. Doesn't let the teenage cell go into the maturity adult cell. And it's as long as it's in this state, the cell doesn't progress. But once it gets tagged by some enzymes with some phosphate groups, what it does, it gets targeted to another protein, and that protein puts some other tags on it, these U tags, that means it's tagged for degradation. It's called ubiquity. So once the cell gets rid of SIC1, now it's ready to progress and divide and so on. Now, if you look carefully what happens with the charge of the protein as it gets phosphate, there is little binding where you add 1 phosphate, 2 phosphates, 3, until you get about 5 or 6 phosphates. Then you get a very sharp increase in binding. So the question here is, why does this protein respond in this sharp response, sharp line to phosphorylation? Is it perhaps charge? Is it something about the dimension? So my colleague, so this is something that my colleagues at UFT, Julie Foreman-Kay and Hussan Chan, studied. And Hussan, a decade and a half ago, came up with this polyelectrostatic model where there's like, think of it as a mean field effect, where the protein never quite leaves a single binding site. And adding phosphates, the protein becomes quasi-neutral, and the protein can relax and collapse a little bit rather than be electrostatically repulsing each other and being rigidified. Now, this protein not only has to be compact, but it also has to stretch a certain gap, which is quite significant. Think of it as, this is about 6 nanometer gap. So if it's bound to its partner, if it's bound by this guy, it also has to stretch pretty far to get these U tags. And how far? It has to be simultaneously both compact due to polyelectrostatics and be tagged by this other subunit for degradation. So the question that the physicist comes like, what experiments, how do we determine these relevant dimensions of this problem? So this is where I came in about a decade ago with the techniques that we use in our lab called this FRET, or resonance energy transfer. We measure distances on this length scale, say angstrom. 10 angstrom is a nanometer, so between 20 and 80 angstrom, we can measure specific distance, and in this case, the end-to-end distance. We need a label. NMR colleagues like Julie Foreman-Kay and other people, they use paramagnetic relaxation, chemical shifts. They need to put either a small label or none, and they get different kind of restraints. These are average distance restraints across the entire chain, but on a much smaller length scale. And last but not least, you also have X-ray scattering, which gets the distribution of intermolecular distance with the radius of gyration. Where is the mass concentrated in this? The beauty of this, it doesn't require any label. The disadvantage of these proteins that require quite a lot of protein, and you may get aggregation. So let me get to the core of this talk. Integrative ensemble modeling. What does it mean? It means to take this information from disparate length and time scales into a richer description of IDP systems. So we will start with a set of all atom structures. I'll tell you in a minute how we generate them. And then we impose these restrictions and we try to fit the back calculated quantities to the observable data. So this machine, this algorithm designed about a decade ago in Julie's lab, called ensemble, not to be confused with conformational ensemble, which is the set of structure. So ensemble with capitals. What does it do? It tags up on this key. You have a Monte Carlo selection from an initial soup until you fit the restraints, and then you spit out a final ensemble. And before we came into the picture, this is a magnetic NMR lab. They may have used SACS a little bit, but they didn't use single molecule fluorescence information. So enter Greg Gomes, a PhD student in my lab, and this is an undergrad. Ashley, they worked on this for 2 or 3 years, and this is a schematic of what we achieved. We had this set of structures, and we swapped them in and out, and we have a number of calculators where we calculate the scattering profile, we calculate local structure, we calculate non-local structure. So all this approach is refining and refining and refining until you start see initially it doesn't fit the experimental observable, and then in the end, the red curve, if you look, it stays within this area, within the uncertainty of the experiments. And the same, to a certain extent, is true for chemical shift, for X-ray scattering. And here what you have is the measured versus the calculated fret. This shows again how we do it for each conformer. We get the scattering profile and we match it to the average experiment. This is in a way, you see the labels are fairly big compared to the proteins. This is our dyes. We let them freely rotate and we look at energy transfer between this tag and that tag. And for each conformers, we get a certain value. We look at the average in this ensemble and we swap in and out conformers until we simultaneously reach agreement, not only with this observable, but with that and 2 others. Okay, this is to show you how the experiments look like. we did measurements on SIC1, on the phosphorylated SIC1, remember with 6 phosphates, and we extract distances, end-to-end distances. We did X-ray scattering on these measurements, and this, pardon me, we extracted radius of gyration as well as probability of internal distances. Let me tell you about the known unknowns in the ensemble modeling. So when you have a multitude of experimental restraints, one could ask, hey, wait a minute. What if they are cross-talking to each other? Does it depend on the order on which we impose these restraints? Maybe we should reweigh them because they don't have as many degrees of freedom as we need. So now the other stuff is that the combination of restraints, if we take any permutation, could act in difficult to predict ways. And in the field, when we were doing this a couple of years back, there was a raging controversy, if you believe, between my camp, the SM Fred camp, and the X-ray scattering camp, saying that each camp does measurements wrong and we do the wrong inference. Now the conflict is solved, but basically that raises the idea, are different experiments mutually consistent, or is there something either due to the label or to the interpretation or to the back calculation that people are doing wrong? So consistency is key. So let me show you how the ensembles that we played with, how did they fare against experimental? So we use this statistical coil generator of trades to generate prior. This is basically no physics other than excluded volume. So we didn't allow these balls and sticks to overlap with each other. So only hard sphere van der Waals repulsion. So when we look at the initial soup, so no weighing whatsoever to the experimental restraints, we got a pretty lousy agreement with paramagnetic relaxation, X-ray scattering. This is the measured Fred efficiency. This was the characterization, quite a big discrepancy. These were fairly good. The chi-square were less than 1, but as you will see in a minute, this seemed not to be extremely sensitive to the choice of conformants. Now, if we impose only SACs, X-ray scattering data is restrained, yes, we get a very good agreement. But look what happens. Fred becomes even worse. The magnetic resonance data becomes even worse. And so do this to a certain extent. We can say, hey, let's impose NMR restraints. Yes, this we get agreement here. But look here, this one blows off the chart. Not only that, but Fred swings all over this and goes to the other. So we get over compact. We go from over expanded to over compact conformation. Now, you would kind of guess, then the best is let's combine the two. And when we impose simultaneous SACs and PRE restraints, look at this. We get agreement here. We get agreement here. These are not too bad either. And boom, the Fred efficiency is very close to what we measured. In addition, if we now really hardwire the chemical shifts, alpha and beta, the two types of carbon, I'm not going to go into detail, we get better agreements here. And guess what? Even the Fred efficiency comes spot on to this. This was not used as a restraint in the ensemble module. What you have in machine learning, we use it as a validation, right? So it's when you have data sets, you keep something that you restrain and you keep something external for validation. So the corollary that came out of this is that at least in our case, either we were lucky or we did beautiful controls. This influence of probe, SACs versus SM Fred discrepancy did not play a role. I want to show you a bit of polymer physics, polymer scaling, but in a nutshell, for both proteins, what we found is that on the small and intermediate scale, the distance between residues, between positions, looked as a good solvent, like your normal homopolymer single exponential. So the scaling factor is 0.5. This is a good solvent. Good solvent means your protein really interacts well with this. So it's well solvated by the environment. So not so much intramolecular, but interaction with the solvent. Look what happens at large separations between the residues, it undershoots. So it becomes like a poor solvent. So the more remote residues are from each other, the more compact. So they're expanded on the short scale and they're contracted on the long scale. And more so surprisingly, more so for the unphosphorylated than for the phosphorylated. Remember when we talked about the polyelectrostatic, we said that upon phosphorylation, our protein gets a little bit more compact, and that's what helps staying bound to the complex with fairly high. We've done some even other types of analysis. We did 2D maps where we look at how intramolecular distances, like say residue 10 to 90. You see blue here. It means that the distance on average between residue 10 and 90, so between the two ends, it's about 0.8 or less than in a random coil. So it's more compacted than a random. And it's true for both SIC1 and phosphorylated version. And when you can even do comparison between the two forms of the protein, and you can see expansion, these aisles of expansion happen to be around the phosphorylation state. Anyway, I won't go into details, but just to give you a flavor how calculating this ensemble is just the first step. The cool part, the biology or even the interesting physics, comes when you do data mining of this ensembles, which consists, by the way, here, what we have 5 ensembles, each with 100 conformers. So we repeated the procedure 5 times so that we got some statistics. So we have 100 conformers in each ensemble. Okay, how am I doing for time? 15 minutes. So what I want to show you here, forget about all this. They're all polymer physics quantities, some ratios between end-to-end distance and radius. This is something about shape, sphericity versus. What I want to point out is that from our calculations, from our analysis, it turns out that the protein has fairly large fluctuation, although it's more spherical, more compact than an excluded volume or some called theta solvent, where interactions with itself and with the solvent are balanced. Although it's more compact, it's also more fluctuating. And what this allows you is to, in a schizophrenic kind of way, simultaneously satisfy this condition of being bound to the target, which is this protein. This is a dimer, so that's why you have a copy. So simultaneously to be bound to this guy and to be able to reach this other part of the complex where it gets tagged for degradation. So you can be compact, but also fluctuate. And it's also good for read-write for this post-translational modification, like phosphorylation and ubiquitination. So a protein doesn't exist in the same state. It has to be available for other enzymes to bind and attach different groups. So where do we want to go from here? We want to evolve this ensemble, and we are now busy with Julie's lab and another group at Berkeley to introduce our data as direct restraints and maybe swap out others as validations, or develop other data as validation. We also are, because we detect kinetic data, we get rates. We also want to introduce temporal information about these interconversion rates into the modeling. And we have a manuscript that we just submitted to Journal of Physical Chemistry about this with Teresa's. And I want to show you in the last 10 minutes or so that we can also use other methods that are a little bit more balanced when it comes to prior versus experiment. So this is called Bayesian maximum entropy method. And we use different priors, molecular dynamics simulation, to generate the initial soup of conformation. So the BME approach, which is designed by Creston, Larsen, and Copenhagen, but I'll give you a reference in a minute. What do we want to, the thought process is like this. You want to integrate diverse experimental data. So you want your model to fit the data accurate. So you want to minimize this quantity, chi-square. M is the number of observations. And sigma is the precision of each experiment, obviously for students. So what I told you so far, we use this very archaic way of generating poor man's way to generate structures when you don't have molecular dynamic simulations. You use this traits, which is a method that generates structures based on excluded volume. And the secondary structure, so helices, beta sheet, extended, so it's kind of a probability based. Now, if you're rich or if you're lucky, you get molecular dynamics to generate the prior. And these molecular dynamics force fields are consistent with physical laws up to a certain approximation. However, the more elaborate prior you have, you wouldn't want to cut through it and to throw it away with the same kind of cavalier attitude that you did with trades, because you knew this was crap, but you want to keep some of this prior. So you cannot just minimize chi-square. So what Creston did, said, okay, what about we try to do both? At the same time, I want to maximize this relative Shannon entropy. So when I reweigh my initial conformation, I have to minimally reweigh them in order to fit the data. So the function that you see here is very analogous to a free energy function, where you have an enthalpic term and an entropic term. And this parameter theta controls the balance between fitting the data and deviating from the prior. So if you have theta equals 0, you basically don't trust the prior. You just fit until you get. You may overfit, but you don't care. When you don't trust the data, you have this other stuff where you have no confidence, so you underfit. The data doesn't matter much in the optimization. So we apply this. This is from a manuscript in preparation, and I want to show you. It's complicated to explain everything, but you have two methods: BME, the Bayesian maximum entropy method, and ensemble, and you have three different priors. Trades is here. So this upper corner, lower corner, trades and ensemble is what I showed you before. We use this prior and we use this method to optimize our system, to get all these quantities in the gray zone and hopefully the threat to land in this blue zone. What we've done here, we have now two molecular dynamics force fields in addition to a new method. So we have 6 different ensembles here. What in the paper, I'm not going to show you, we looked at what unites and what distinguishes each and every of these ensembles to understand, hey, how much does having a good prior, how much does that matter? What can we say about certain intermolecular interactions, like hydrogen bonds or pi interactions? So I want to close in the last 5 minutes, showing you a little bit of data that we measure on an entirely different system, disorder system. It's called 4EBP2. It's involved in the neurons. It's involved in learning and memory, in the synaptic plasticity, and it acts again like a gatekeeper for what? For initiating translation. Translation in biology means making proteins, converting RNA to proteins. So you see, when it gets phosphorylated, this protein doesn't bind to the target. It's released from the target, and the activator can bind. So again, my collaborator Julie, and we also involved in this, showed that the disorder protein in the non-phosphorylated form has a super high affinity to bind to this target. So look at this. The lower the number, the tighter the binding. So you have a disorder protein, and it wraps around this target. Now, if you phosphorylate it, what it does, it folds and it detaches. So it goes into this state where the Velcro gets hidden. So it's sequestered inside this beta fold. And it's basically what we showed is even the remainder of the protein, this disordered part in the C-terminus, it's also contributing to further weakening this interaction. Now, you may ask, this is weird. I thought that disordered proteins, if they fold, they may have a higher chance to interact with their partners. In this particular case, it looks like the opposite. The phosphorylation leads to folding, and the folding leads to release or unbinding. So what did we do here? We put tags. This is the sequence of the protein. There is about 120 letters Don't memorize them all. It shows where phosphates are added. But look at the blue and the red spots. This is where we engineered tagging positions, where we put our blue and red tags to probe distances, intramolecular distances in this. So you will have one construct that probes partially the folded domain, once phosphorylation, and we have another construct, 73 to 121, that only looks at the disordered C-terminus. So Spencer and Jianfu, what they did these measurements, and look at this beauty here. So typically, this is the non-phosphorylated version of the protein, 3291. We measure a certain fret value, a certain distance. Hey, when you fold a protein, you typically compact it. So if we expected that upon phosphorylation, when the protein goes from disordered to folded, the distance between the probes will decrease, which means that the fret efficiency should go to the right. However, we found that it goes to the left. And not only that, but also still remains, if it's on the red curve, it means that it's flexible, dynamic. If it would be on the black curve, it would mean that it's as rigid as a rod, like a DNA kind of rigidity. Look at this. Even when it's bound to its target, to this 4E protein, what our measurements indicate, we go even further apart. These probes go even further apart, and now we get some sort of stiffening of the chain. Now, when we look at this C-terminus, the trend is much less. But what this shows is that we have still a very dynamic and dynamically engaged part of the protein with the target. I'm not going to show you the rest of the data, but the paper which we just submitted shows a number of experiments that we performed. And the main finding was this, that the C-terminus of the protein that was kind of neglected, think of this guy here. This part really plays a significant role in interacting with the protein. Not only these two domains, this bipartite dynamic interface, but the C-terminus does really play a role in the affinity of. So it's a tripartite, highly dynamic binding model. So want to show Thomas, he applied BME, and we're now doing ensemble optimization. This time BME allows to import fret as restraints. So you see we have chemical shifts, we have X-ray scattering, we have two distance restraints, pretty cool. However, BME doesn't have a well-designed module for PRE. So what we do here, we use the paramagnetic relaxation as external validation. So as you see, as theta decreases, we get better and better agreement with the experiment. And we can use the so-called the knee or the elbow method to find out where to stop, because you could go on and on and on and discard most of your prior. So right now we're data mining these ensembles that Thomas generated on based on our recently measured data. So this brings me to my conclusion, which is perfect for 58. What did I show you? I showed you that we can generate finite conformational ensembles for two what I would call prototypical or emblematic IDPs. We're lucky that there's a rich amount of structural data on these disordered proteins. How did we do it? By implementing complementary data, using validations, but also moving towards new methods like BME. I hope that I briefly show how the properties that we derived from these ensembles that we calculate are congruent, are consistent with biological functions. And the case that I'm making is the integrative use of what? Multiple experiments, probing disparate scales. We use full computational modeling, although I may claim that I'm playing like an imposter here. I'm an experimentalist, talking about computational modeling. And I even claim to know something about polymer physics. So with that, I just want to give credit to the people that did the work, primarily Greg Gomes, now a postdoc at Yale, and soon to be a research scientist at this anti-aging institute on the Silicon Valley. Spencer Smythe is here. Past grad student Jian Fu worked a lot in Baoshu. Two talented undergrads, Ashley and Thomas, worked. Plus, I was blessed with very good collaborators, Julie Foreman Kay at Sick Kids, and more recently, Teresa had Gordon at Berkeley. And that I'm thanking you for your attention, and I leave you with this I'll let you read this. Physics is difficult but simple, and biology is easy but complex. So thank you for your attention. I'll be happy to take